Welcome back to our channel. Today, we continue our journey through the fascinating world of the animal kingdom with part 3, focusing on two unique phyla, Ctenophora and Platyhelminthes. If you haven't seen part 1 and part 2, be sure to check them out first. You'll find the links in the description and in the cards above. For a structured learning experience, don't forget to explore the full Animal Kingdom playlist, also linked below and in cards too. As always, detailed notes and resources are available through our Linktree, all for your quick revision and NEET preparation. Let's dive in and discover the wonders of biology together. Before we dive deep into the concept, make sure to refer to the official syllabus and previous year questions, PYQs, from here. These resources will give you a clear picture of what to expect in the NEET exam and help you focus on what truly matters. Understand the weightage. Identify high-yield topics. Learn from PYQ trends. Stay ahead with smart preparation. Remember, smart work begins with knowing what to study. So, take a moment to explore the syllabus and PYQ list, and let's build a strong foundation together. Let's revise the broad classification of the animal kingdom. First, based on the level of organization, the simplest animals, like Porifera, show a cellular level of organization. From Coelenterata onwards, animals show higher levels like tissue, organ, or organ system level of organization. Next, we classify animals based on symmetry. Porifera are asymmetrical, meaning they have no definite shape. Animals like Coelenterata and Ctenophora show radial symmetry, where body parts are arranged around a central axis. From Platyhelminthes to Chordata, animals show bilateral symmetry, which means the body can be divided into mirror halves along one plane. Now let's talk about the body cavity, also called the coelom. Acoelomates have no body cavity. An example is Platyhelminthes. Pseudocoelomates have a false coelom that's not fully lined by mesoderm. Asculmints are examples. Coelomates have a true coelom completely lined by mesoderm. Examples include Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata. Here's an important note: Echinodermata have bilateral symmetry in the larval stage, but show radial symmetry in the adult stage. Refer to the images for more clarification. Let's quickly revise the differences between two animal phyla, Porifera and Coelenterata. Members of phylum Porifera, commonly known as sponges. They are mostly marine, with some found in freshwater. They exhibit asymmetrical body symmetry. Their body organization is at the cellular level, meaning they lack true tissues and organs. A unique feature of sponges is their canal system, which allows water circulation throughout the body. They do not have a body cavity, and digestion takes place intracellularly. Their skeleton is composed of spicules or spongin fibers. Specialized cells called choanocytes or collar cells help in feeding and circulation. Sponges reproduce asexually through fragmentation, while sexual reproduction is hermaphroditic, with internal fertilization and indirect development. They do not exhibit distinct polyp or medusiforms. Examples of Porifera include Cycon, Spongilla, and Euspongia. On the other hand, phylum Coelenterata, also known as Cnidaria. It includes aquatic animals that are mostly marine. It shows radial symmetry. They have tissue-level organization and are diploblastic, meaning their bodies develop from two primary germ layers. A key feature of cnidarians is the presence of cnidocytes, or stinging cells, on their tentacles. They possess a gastrovascular cavity with a single opening that functions as both mouth and anus. Digestion occurs both extracellularly and intracellularly. In corals, the skeleton is made of calcium carbonate. Their specialized cells are cnidocytes, which contain structures called nematocysts. Asexual reproduction in cnidarians commonly occurs through budding, especially in the polyp form. Sexual reproduction can be external or internal, and many species show metagenesis, which is the alternation between polyp and medusa stages. Examples include Phasalia, Adamsia, Panatula, Gorgonia, and Meandrina. Let's study about Phylum Ctenophora, which is also known as comb jellies or sea walnuts. Includes organisms that are exclusively marine. They are radially symmetrical. They are diploblastic, with a tissue level of organization. A unique feature of ctenophores is the presence of 8 rows of ciliated comb plates that help in locomotion. They also exhibit bioluminescence, the ability to produce and emit light. Digestion in ctenophores occurs both extracellularly and intracellularly. These organisms are hermaphroditic, meaning sexes are not separate. Fertilization is external. Development is indirect, involving a larval stage. Examples include Pleurobrachia and Ctenoplana. Refer to the images for clarification. Now, let's talk about Phylum Platyhelminthes, which is commonly known as flatworms. They have a dorsoventrally flattened body. They are mostly endoparasitic, living inside animals and humans. These organisms show bilateral symmetry. They are triploblastic with 3 germ layers. They are acoelomate, meaning they lack a true body cavity. They exhibit an organ level of organization. Parasitic forms have hooks and suckers for attachment, and some absorb nutrients directly through their body surface. Flame cells help in excretion and osmoregulation. Planaria shows remarkable regenerative ability. Flatworms are hermaphroditic, with no separate sexes. Fertilization is internal. Their development occurs through multiple larval stages. Examples of platyhelminths include Taenia, commonly known as tapeworm, Fasciola, also called liver fluke, and Planaria, often referred to as cross-eyed worms due to their eye spots. Refer to the images for clarification. Let's quickly revise Phylum Ctenophora and Platyhelminthes. Let's begin with ctenophores, which is also known as sea walnuts or comb jellies. While platyhelminthes are flatworms, ctenophores are marine animals. Flatworms are mostly parasitic, found in animals and humans. Ctenophores show radial symmetry, whereas platyhelminthes show bilateral symmetry. Ctenophores are diploblastic, while platyhelminthes are triploblastic. Ctenophores lack a coelom, while platyhelminthes are acoelomates. Tissue-level organization is seen in Ctenophora, but organ-level in platyhelminthes. Ctenophores move using 8 ciliated comb plates, while flatworms show limited or no movement, especially in parasites. Both have incomplete digestive systems. But ctenophores exhibit both extracellular and intracellular digestion. A special feature of ctenophores is bioluminescence. In flatworms, flame cells perform osmoregulation and excretion. Regeneration is not observed in ctenophores, but is highly developed in Planaria. Both are hermaphrodites, meaning sexes are not separate. Fertilization is external in ctenophores, while internal in platyhelminthes. Development is indirect in both, with larval stages present. Examples of Ctenophora include Pleurobrachia and Ctenoplana. Examples of platyhelminthes include Taenia, Fasciola, and Planaria. Let's practice few questions to test your knowledge. Question 1. Which of the following is a feature of flatworms, platyhelminthes? From NEET 2022. Option A, pseudocoelomate. Option B, triploblastic and acoelomate. Option C, diploblastic and coelomate. Option D, triploblastic and pseudocoelomate. Explanation. Platyhelminthes, also called flatworms, are triploblastic, meaning they have 3 germ layers, ectoderm, mesoderm, and endoderm. However, they lack a true body cavity, making them acoelomate. Pseudocoelomates have a cavity not fully lined by mesoderm, like ascomynths. Diploblastic is seen in lower phyla like Cnidaria. The correct answer is option B, that is feature of flatworms, platyhelminthes, is triploblastic and acoelomate. Question 2. Hooks and suckers are present in which of the following organisms? From NEET 2020. Option A, Planaria. Option B, Fasciola. Option C, Taenia. Option D, Hydra. Explanation. Taenia, or tapeworm, is a parasitic platyhelminth that attaches to the intestinal walls of its host using hooks and suckers on its scolex or head. Planaria is free-living and lacks hooks or suckers. Fasciola, a liver fluke, has suckers but no hooks. Hydra belongs to the phylum Ctenophora and does not have these attachment structures. The correct answer is option C, that is hooks and suckers are present in Taenia. Question 3. Which of the following phyla shows flame cells involved in excretion and osmoregulation? From NEET 2016. Option A, Porifera. Option B, Platyhelminthes. Option C, Cnidarian. Option D, Mollusca. Explanation. Flame cells are specialized excretory cells found in platyhelminthes. They help in osmoregulation and excretion by removing excess water and nitrogenous wastes. Porifera lack any excretory organs. Cnidarians are too simple to have such systems. Mollusks use nephridia instead. The correct answer is option B, that is platyhelminthes shows flame cells involved in excretion and osmoregulation. Let's move to question 4. Which phylum is exclusively marine and exhibits bioluminescence? From NEET 2023. Option A, Porifera. Option B, Coelenterata. Option C, Ctenophora. Option D, Mollusca. Explanation. Ctenophores, also known as comb jellies, are exclusively marine animals that exhibit bioluminescence, the ability to produce light. This light emission helps in defense and communication. While some coelenterata may also glow, it's not a common trait across the group. And Mollusca do not show bioluminescence as a defining feature. The correct answer is option C, that is Ctenophora is exclusively marine and exhibits bioluminescence. Question 5. Ctenophores differ from cnidarians in having: Option A, nematocysts. Option B, bioluminescence and comb plates. Option C, polymorphism. Option D, flame cells. Explanation. Planaria is a type of flatworm from the phylum Platyhelminthes, best known for its extraordinary regeneration abilities. It shows bilateral symmetry, is triploblastic, and lacks a true coelom. In contrast, annelids are segmented worms like earthworms, nematodes are unsegmented roundworms, and coelenterata include aquatic animals like jellyfish and corals. The correct answer is option B, that is ctenophores differ from cnidarians in having bioluminescence and comb plates. Question 6. Which of the following is a triploblastic, acoelomate, bilaterally symmetrical organism? Option A, Hydra. Option B, sea anemone. Option C, Taenia. Option D, Aurelia. Explanation. Ctenophores pose of comb plates that beat in coordination for movement, and they are naturally bioluminescent. Unlike cnidarians such as Hydra and jellyfish, which have nematocysts or stinging cells, ctenophores lack these structures. Flame cells are absent in both groups. Also, while polymorphism is common in coelenterata, it's not a feature of ctenophores. The correct answer is option C, that is Taenia is a triploblastic, acoelomate, bilaterally symmetrical organism. Thanks for watching. If you found this helpful, don't forget to like, share, and subscribe to support the channel. Follow us on all our social media platforms so you never miss an update. We're posting daily MCQs, PYQs, and concept breakdowns just for you. Be sure to check out the playlist for this chapter and our daily PYQs and MCQs series to keep your NEET prep strong. More parts of this chapter are on the way, so stay tuned, stay consistent, and stay confident. See you in the next video. Let's crack NEET together.